So now I will be starting with the fungi. Study of fungi, it is known as mycology. Fungi is a unique group of eukaryotic organisms which are heterotrophic in nature. Unique group having eukaryotic heterotrophic organisms. This group shows great diversity with respect to the morphology as well as habitat. Great diversity is seen with respect to morphology as well as habitat. So first of all, I will be dealing with the habitat. Fungi is cosmopolitan in nature, present almost everywhere. Cosmopolitan है, almost present everywhere. Fungi can be both aquatic as well as terrestrial. Aquatic भी हो सकती है, terrestrial भी हो सकती है, but most of the fungi, they are mainly terrestrial. Most of the fungi, they are terrestrial in nature. Only few, they are aquatic. अगर fungi tree bark के ऊपर grow कर रही है, if the fungi is growing on tree bark, this type of fungi, it is known as corticolous. Yes, so you are going to call it as corticolous. अगर fungi grow कर रही है over dung, fungi is growing on dung. This fungi, it is known as coprophilous. Coprophilous fungi. अगर fungi grow कर रही है wood के ऊपर, fungi is growing over wood, then known as epixylic. If fungi is growing over burnt wood, yes, growing over burnt wood, then it is known as xylophilous. Last one, if fungi is growing over hairs, thorns, then fungi is known as keratinophilous. Right? So you can see great diversity is there with respect to habitat of fungi. Now after the habitat, now I will be dealing nutrition in fungi. Nutritionally, they are heterotrophic organism. Heterotrophic nutrition is there. In heterotrophic nutrition, they can be parasitic. They can be symbiotic. They show symbiotic association both with the higher plants as well as lower plants. Symbiotic association both with the lower plants as well as higher plants. They form symbiotic association with the lower plants, forming lichens. You might be knowing lichens is a symbiotic association between algae and fungi, lower plants and fungi. They can show also association with higher plants like gymnosperms, angiosperm, and this association which is formed, this is known as mycorrhiza. This is mycorrhiza. Lichens and mycorrhiza I will be dealing in detail in this chapter laterly. Along with this, there is also presence of saprophytic nutrition, which is very well known to you, that is obtaining food from dead and decaying organic matter. 3 तरह का heterotrophic nutrition fungi के अंदर present है. But among these 3 categories, which is the main one? Among these 3 categories, main type of nutrition which is present, that is saprophytic nutrition. Saprophytic nutrition, fungi, they are major decomposers along with the bacteria. Along with the bacteria, major decomposer which are present in the soil, that is fungi. But fungi generally prefers to grow in acidic condition, and bacteria prefers to grow in alkaline neutral condition. So fungi generally prefers to grow in acidic soil. That's why you can call it as acidophilic in nature. Now after looking into the nutrition, now I will be dealing with body structure of fungi. Body structure. Body structure can be unicellular. One example which is very well known to all of you of unicellular fungi, that is yeast. Along with that, body structure can also be multicellular. जो मल्टीसेलुलर फंजाई है, इस फंजाई के अंदर बनता है फिलामेंटस ब्रांच्ड स्ट्रक्चर. दिस फॉर्मेशन ऑफ फिलामेंटस branched structure. You know this filamentous branch structure which is formed, this is known as hyphae. बहुत सारे hyphae aggregate करेंगे. These hyphae, they will aggregate forming tissue-like structure. The hyphae, they will aggregate forming tissue-like structure. This tissue-like structure which is formed, that is mycelium. ये सिर्फ एक tissue-like structure है, but it is not a true tissue. That's why mycelium is also known as loose tissue. Right? Now this mycelium can be of several types. माइसीलियम के अंदर ये मैं फिलामेंट दिखा रही हूं. इन द फिलामेंट ये मैंने दिखाए हैं न्यूक्लियाई, और साफ नजर आ रहा है आपको यहां पर कि इस फिलामेंट के अंदर कोई भी सेप्टा नहीं है. इन द फिलामेंट देयर इज नो सेप्टा. इन द फिलामेंट देयर इज नो पार्टीशन. इन द फिलामेंट देयर इज नो सेल वॉल. बीच में कोई भी सेल वॉल पार्टीशन नहीं है. Such type of filament where cell wall is absent, septa is absent, it is known as aseptate mycelium. Aseptate mycelium, or it can also be known as sinocytic mycelium. Now the second condition, सेकेंड कंडीशन के अंदर अब आप देख रहे हैं कि सेप्टा प्रेजेंट है. Now the septa is present. Septa present है, so now you can simply call it as septate mycelium. In each cell there is presence of only one nucleus. When in each cell there is presence of only one nucleus, this type of condition, this type of mycelium, it is known as primary mycelium. इसको हम कहेंगे प्राइमरी माइसीलियम, और इट इज ऑल्सो नोन एज मोनोकैरियोटिक माइसीलियम. हर एक सेल के अंदर एक एक न्यूक्लियस, एक एक कैरियन, सो इट इज नोन एज मोनोकैरियोटिक माइसीलियम. लेट्स सी व्हाट हैपेंस इन द नेक्स्ट कंडीशन. अगेन सेप्टेट माइसीलियम, पर अब हर एक सेल के अंदर 2 न्यूक्लियाई, हर एक सेल के अंदर 2 न्यूक्लियाई है. दिस माइसीलियम इट इज नोन एज सेकेंडरी माइसीलियम. If the 2 nuclei they are present, it is also known as dikaryotic mycelium. Dikaryotic mycelium is represented as N plus N and primary mycelium as N. ये जो मैंने यहां पर पार्टीशन बनाया है, what is this partition? Septa. So now I will be dealing with type of septa. Types of septa. Three categories, 3 types are there. First condition, ये मैंने septa बनाई है. I've made this septa. If in the septa no distinct pores they are present, सेप्टा के अंदर डिस्टिंक्ट पोर्स एब्सेंट है, दिस टाइप ऑफ सेप्टम इट इज नोन एज कंप्लीट सेप्टम. यस, सो यू आर गोइंग टू कॉल इट एज कंप्लीट सेप्टम. सेकेंड कंडीशन, now you can see in the septa there is presence of one central pore, एक central pore present है, and this pore is blocked, it is plugged, some part of the pore is plugged with the help of this structure. This structure is known as Voronin body. Some part of the pore is plugged with the help of Voronin body. Around Voronin body there is presence of plasma membrane. Around the Voronin body there is presence of membrane. Membrane present है. This type of septa where there is presence of single central pore, it is known as simple pore septum. Right. Now last category, this is last category. In the last category you can see कि पोर के आसपास जो सेप्टा है, दैट सेप्टा इज स्वॉलन. अराउंड द पोर सेप्टा इज स्वॉलन, फॉर्मिंग बैरल लाइक स्ट्रक्चर. इस सेप्टा के ऊपर, above the septa there is formation of a cap. सेप्टा के ऊपर एक कैप है. Below septa also there is presence of a cap. This cap which is present, this is known as parenthosome. And this cap is made up of endoplasmic reticulum. This cap is made up of an organelle, membrane-bound organelle, that is endoplasmic reticulum. This type of septa, it is known as dolipore septum. Yes, this is known as dolipore septum. So this was all about body structure. Body structure के अंदर हमने पढ़ा that fungi can be unicellular, multicellular, can be septate or aseptate. पर इन सारी forms में से जो सबसे common form है, mainly in the fungi there is presence of septate branched mycelium. This is main type present in fungi. Now after body structure, I will be dealing with cellular structure. Now cellular structure. Outermost covering, that is cell wall. Cell wall is made up of chitin. Cell wall, it is made up of a polysaccharide, that is chitin. Chitin is polymer of NAG. NAG, that is N-acetyl glucosamine, which we have studied also in the bacteria. तो ये हो गई outermost covering, that is cell wall. अब cell wall के बाद I am forming cell membrane. This is cell membrane. This is cell membrane, and you can see here outgrowth of cell membrane. These are outgrowth of cell membrane. Outgrowth of cell membrane, they are known as lomasomes. In the case of bacteria, we have studied ingrowths of cell membrane. They are known as mesosomes. These are outgrowth. They are known as lomasomes. Now the function of lomosome is that they increase surface area for transport of compounds. Increase surface area for transport. Now this is cytoplasm. In the cytoplasm there is presence of membrane bound organelles. One of membrane bound organelle which can be present, that is Golgi apparatus. Golgi apparatus. In the Golgi apparatus, in the Golgi bodies, there is presence of sac-like structure. ऐसे sac-like structures present होते हैं. ये जो sac-like structure present है Golgi bodies के अंदर, इनको हम कहते हैं cisternae. And in the fungi, this Golgi bodies, the Golgi apparatus consists of only one cisternae. Golgi apparatus consists of only one cisternae. So you can call it as unicisternal condition. Only one cisternae is present in Golgi bodies. Now after this, we can see reserve food. Reserve food which is present, that is glycogen and oil. Along with that, in the case of fungi, there is presence of special type of karyokinases. In the fungi, there is presence of special type of nuclear division. This nuclear division, it is known as karyokarosis. जो नॉर्मल न्यूक्लियर डिवीजन होती है उसको हम कहते हैं कैरियोकाइनेसिस. यहां पर स्पेशल टाइप ऑफ न्यूक्लियर डिवीजन है, दैट इज नोन एज कैरियोकारोसिस. So let's see how karyokarosis is different from karyokinesis. Right. So this is normal one, nuclear division, and this is special one in fungi. Let's see what are the differences. In normal nuclear division, let's see what will happen. ये मैंने बनाया है nucleus. Nucleus is present inside our cell. ठीक है? Let's say this is cell membrane and this is nuclear membrane. Nucleus के अंदर there is presence of genetic material. Genetic material present है in the form of thin thread-like structures. Genetic material present in the form of thin thread-like structure is known as chromatin. This is known as chromatin. जब तक न्यूक्लियस डिवाइड नहीं करता, genetic material is present in the form of thin thread-like structure which is known as chromatin. अब जब न्यूक्लियर डिवीजन होगी तो क्या होगा? Now first of all, this nuclear membrane will disappear. न्यूक्लियर मेम्ब्रेन डिसअपियरेंस होगी. साथ साथ ये जो क्रोमैटिन है ये कंडेंस करेगा. Chromatin will show condensation. After the condensation, there is formation of rod-like structure which are known as chromosome. For simplicity, I am forming only one chromosome here. Chromatin will condense, forming rod-like structure. These rod-like structure which are formed, they are known as chromosome. Now what will happen next? Now there is formation of spindle fibers. इसके बाद बनेंगे स्पिंडल फाइबर्स. और ये जो स्पिंडल फाइबर्स है इस क्रोमोसोम को स्प्लिट कर देंगे so that one part of the chromosome can move to one pole, other part of the chromosome can move to other pole. तो जो नॉर्मल कैरियोकाइनेसिस होता है वहां पर न्यूक्लियर मेम्ब्रेन डीजेनरेट करेगी and this formation of spindle. The spindle which is formed, that is not inside the nucleus. Nuclear membrane degenerate होने के बाद बन रहा है, so it is known as external spindle. Now what will happen in karyokarosis? In the karyokarosis, nuclear membrane will retain. Nuclear membrane retain रहेगी. Inside the nucleus, now the chromatin will form chromosome. After condensation, formation of chromosome is there, and again formation of spindle. तो अब जो स्पिंडल बन रहा है वो न्यूक्लियस के अंदर बन रहा है. Now the spindle is formed inside the nucleus, so you can call it as internal spindle. Formation. So this is very much clear how karyokinesis is different from karyokarosis in fungi. In fungi, during nuclear division, nuclear membrane will persist and the spindle is formed inside the nucleus which is known as internal spindle formation. Now after dealing with cellular structure, now I will be dealing with classification of fungi.